Today I'll discuss a malgodynamic simulation study of protein sequencing through the nanopore. The motivation behind this study is the success of the Oxford nanopore sequencing. This technology realized the DNA sequencing by utilizing the nanopores and nanometer size proteins and DNA. For this study, we have considered a bilayer graphene sheet with the pore size is the 2 nanometers. I have engineered a peptide sequence in which first 8 amino acids are the arginine and the last 5 are the homo sequence of the any amino acids among the 20 standard amino acids. My simulation box, initially simulation box dimension is the 5.3 cross 5.3 cross 20 nanometer cubes, and I have applied the electric. I have applied. It's okay. I have applied electric field 0.05 volt per nanometer in the positive z directions. All the simulation conducted by using the GROMAC softwares with the force field charm 36 modified. To achieve the statistically meaningful data, we have performed 250 simulations for each amino acids, in which some of the trajectory was discarded because of the translocation events. We have calculated the dwell times and the blockade currents for these simulations. The dwell time is basically the time duration between the initial entry of the first residue of the histidide sequence into the pore and the subsequent exit of the last residue. Basically, you can say this is the time spent by the histidide sequence inside the pore. Here you can, in this video, you can see clearly see the what is the residence times I am calculating. This T is equal to T1 when the first residue of the peptide is going inside the pore, and the T is equal to T2 when the last residue of the peptide, and the residence time is the T2 minus T1. Oh, it's not working. So I have also calculated the blockade currents for using this equation, and for the two cases. So the first case is the when no peptide positioned within the pores, and this is the baseline currents, or you can say the open pore currents, and its value is the 6.86 plus minus 0.2 nanoamperes. I have also calculated the current when the peptide is positioned within the pores, and this is denoted by the I. I have also defined a blockade current equal to I0 minus I divided by the I0. This is my open pore characteristic. Here you can see the IV curves follow the ohmic behaviors, and the calculated pore conductance is the 7.07 nano Siemens, and this is the good agreement with the experimental calculation. I have generated a KDE plots by using the 200 data points extracted from the 200 trajectories, and I have also extracted the most probable value of the dwell times and the blockade currents. For the positively charged amino acids, here you can see. For the positively charged amino acid, here you can see the dwell times and the blockade currents for the arginine is the highest. This is the generally because of the van der Waals volume of the arginine is very high. Again, here you can see the van der Waals volume of the protonated histidine is the less than from the lysine, but they have comparable dwell times and blockade currents. This is because of the ring structures present in the protonated histidines. So we can see the size and the spatial characteristic of the amino acid residues play a crucial role in determining their behavior within the pore and their impact on the blockade current. This is the probability distribution of the dwell times and blockade current for the protonated histidines and the non-protonated histidines. Here you can see the significant difference in the dwell times, and this suggests the protonated has a notable effect on the translocation dynamics. Here you can see the dwell times. The dwell time difference is very high in the case of the protonated and non-protonated histidine. This is just because of the action of the electric fields on the positively charged protonated histidines. But here you can see the blockade currents are the similar. Yeah, you can say this is because of the similar van der Waals volume of the protonated and non-protonated histidine. For the polar uncharged amino acids, here you can see the dwell times for the GLN is high. It's again just because of the van der Waals volume of the GLN is high. And but here you can see the van der Waals volume of the ASN and the THR is the similar. And their Blackhead current is the consistent with their similar van der Waals volumes. But you can observe a significant differences in their dwell times. One thing is very important here. You can see the serine has the smallest van der Waals volume in this group, but it has a high dwell times than the ASN. Again, for the non-aromatic types of the amino acids, here you can see the van der Waals volume of the ILE, LU, and MET are the similar, but and the Blackhead current is consistent with their similar van der Waals. But again, here you can see a huge difference, significant difference in their dwell times. One thing is very important here. You can see the van der Waals volume of the MET is less than the similar to the LEU, but the dwell time is higher than the LEU. I have also calculated the probability distribution for the some of the amino acids which do not belong to the any groups. They are the CYS, GLY, HIS, and PRO. Here you can see the CYS exhibit the highest dwell times despite its lower van der Waals volume. The presence of the thiol group in CYS may leads to increase their dwell times. Here you can say, you can say here, you can correlate their Blackhead currents and the van der Waals volumes, and this is the positive correlation. So now we can calculate in the one lines in whole of the simulations. You can say in one lines the Blackhead the dwell time is the more appropriate quantity to distinguish the amino acids as compared to the Blackhead currents.
So when you have these Blackhead currents, since you are trying to make a sequencing of the protein, so is it like a benchmarking? Like, I mean, this amino acid can produce this much Blackhead current and so on. Are they really distinguishable? So I can really making the sequencing accurate? Is it
No,
possible?
actually, we cannot benchmark any Blackhead currents. We cannot set any value, but we can just create an order on the basis of our Blackhead currents.
But
so my Blackhead current shows the same trends as the many researchers has already produced, but here I'm giving the order on the basis of my dwell times, because some of the van der Waals volumes are the similars, so their Blackhead current are the similars. So we cannot distinguish.
So that's what I'm saying. So the sequencing would not be possible, right? If the Blackhead currents become similar for different residues.
Yeah, but the dwell times are the different, because the dwell time is totally depends on the interaction of the amino acid.
So do you have kind of a some kind of map like in the plane of dwell current and Blackhead current, so that you can say this region, this belongs to this residue, and so forth, so you can really do a sequencing?
Yeah, we can do. I think.
And that's unique.
Yeah, this.
I suppose that should depend on the kind of material that you're using, right, for measuring those quantities.
I have done this type of the calculation, but here the time is the shortest, so I cannot.
Thank you.
So the potential application of these findings is to identify the amino acid sequences, right?
Sorry?
The potential application of these MD simulation results is to identify the
amino acid sequences.
Yeah. Okay.